Transgenic Animals. Welcome back to our channel. In today's video, we'll be diving into the fascinating world of transgenic animals. So, grab a cup of coffee, and let's get started. Transgenic animals are created by altering their genetic composition through the addition of exogenous DNA, also known as a transgene. This process allows scientists to study the effects of specific genes and their functions in a controlled environment. There are several methods used to create transgenic animals, but today we'll focus on the three most common techniques used to create transgenic mice. The first method is the retroviral vector method. This involves using a retrovirus to infect cells of an early-stage embryo. The retrovirus carries the desired transgene, which is then integrated into the animal's genome. While this method allows for efficient gene transfer, it has limitations such as short transgene capacity and the potential for helper virus integration. The second method is the DNA microinjection method. In this technique, a clone gene is directly injected into the nucleus of a fertilized egg. The injected eggs are then implanted into a female mouse, and some of the offspring will carry the transgene in all of their cells. While this method allows for the introduction of larger transgenes, the location of integration is random. The third method is the engineered embryonic stem cell method. This technique involves the use of embryonic stem cells that are pluripotent and can differentiate into germ-line cells. The transgene is introduced into these cells through site-specific recombination. The transfected cells are then inserted into blastocysts, which are implanted into foster mothers. This method allows for the establishment of cell lines carrying the transgene on a specific site. Transgenic mice have revolutionized scientific research and have numerous applications. They serve as valuable model systems for studying the biological basis of human diseases and developing treatments. These whole animal models simulate the onset and progression of various diseases, providing valuable insights into their mechanisms. Let's take a closer look at one specific example. Transgenic mice have been created to model Alzheimer's disease, a neurodegenerative disorder characterized by the accumulation of amyloid plaques in the brain. By introducing mutations found in some families with early-onset Alzheimer's, researchers can study the disease's progression and potential treatments. Another exciting application of transgenic animals is the conditional control of cell death. This allows researchers to induce cell death at specific times and under defined conditions in a particular organ. By studying how tissues and organs recover from various degrees of cell loss, scientists can gain valuable insights into organ failure and regeneration. In conclusion, transgenic animals have revolutionized scientific research, providing valuable insights into the genetic basis of various diseases and potential treatments. They serve as powerful tools for studying gene functions and their effects in a controlled environment. Thank you for joining us today on this exciting journey into the world of transgenic animals. If you enjoyed this video, don't forget to hit that like button and subscribe to our channel for more fascinating content. Until next time, stay curious and keep exploring.
Pharma